வணக்கம். வெல்கம் டு NCERT Biology Reading. NCERT Class 11, Chapter 15, Plant Growth and Development. 15.4 Plant Growth Regulators. 15.4.1 Characteristics. The plant growth regulators PGRs are small simple molecules of diverse chemical composition. They could be include compounds. They could be indole compounds, indole 3 acetic acid IAA, adenine derivatives N6 furfuryl aminopurine, kinetin derivatives of carotenoids abscisic acid ABA, terpenes gibberellic acid CGA3 or gases ethylene C2H4. Plant growth regulators are variously described as plant growth substances. Plant hormones are phytohormones in literature. The PGRs can be broadly divided into two groups based on their functions in a living plant body. One group of PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are also called plant growth promoters, example auxin, gibberellins, and cytokinins. The plant growth regulators of the other group play an important role in plant responses to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. 15.4.2 The discovery of plant growth regulators. Interestingly, the discovery of each of the five major group of PGRs have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptile of canary grass respond to an unilateral illumination by growing towards the light source, phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Figure 15.10. Auxin was isolated by FW went from tips of coleoptiles of oat seedlings. The Bacchanaceae disease of rice seedlings, that is foolish seedling, Bacchanaceae foolish seedling disease of rice seedlings was caused by fungal pathogen Gibberella fugicori. Kurosawa, 1926, reported the appearance of symptoms of disease in rice seedlings when they were treated with sterile filtrates of fungus. The active substances were later identified as gibberellic acid. F. Skoog and his co-worker observed that from the internodal segments of tobacco stems, the callus, a mass of undifferentiated cells, proliferated only if an addition of auxin. The nutrition medium was supplemented with one of the following extracts of vascular tissues: yeast extract, coconut milk, or DNA. Miller et al. 1955 later identified and crystallized the cytokinesis promoting active substance that they termed kinetin. During mid-1960s, the independent researchers reported the purification and chemical characterization of three different kinds of inhibitors: inhibitor B, abscission 2, and dormin. Later, all the three were provided to be chemically identical. It was named as abscisic acid ABA. H. Cousins, 1910, confirmed the release of a volatile substance from ripened orange that hastened the ripening of stored unripe bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effects of the five categories of PGRs in the next section. 15.4.3 Physiological effects of plant growth regulators. 15.4.3.1 auxin. Auxins from Greek auxin to grow was first isolated from human urine. Then the term auxin is applied to the indole 3 acetic acid IAA and to other natural and synthetic compounds having certain growth regulating properties. They are generally produced by growing apex of the stems and roots, from where they migrate to regions of their action. Auxins like IAA and indole butyric acid IBA have been isolated from plants. NAA naphthalene acetic acid and T4D 2,4-D 2,4-dichlorophenoxyacetic acid are synthetic auxins. All these auxins have been used extensively in agricultural and horticulture practices. They help to initiate rootling in stem cuttings, an application widely used for plant propagation. Auxin promote flowering, example in pineapples. They help to prevent fruit and leaf drop at early stages but promote the abscission of older mature leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of the lateral axillary buds, a phenomenon called apical dominance. Removal of shoot tips decapitation usually results in growth of lateral buds, figure 15.1, 15.11. It is widely applied in tea plantation, hedge making. Can you explain why? Auxins also induce parthenocarpy, example in tomatoes. They are widely used as herbicides. 2,4-D widely used as kill dicotyledonous weeds. Does not affect mature monocotyledonous plants. It is used to prepare weed-free lands by gardeners. Auxin also controls xylem differentiation and helps in cell division. 15.4.3.2 Gibberellins. Gibberellins are another kind of promotory PGR, plant growth regulator. There are more than 100 gibberellins reported from widely different organs such as fungi and higher plants. They are denoted as GA1, GA2, GA3, and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remains the most intensively studied from all GAs are acidic. They produce a wide range of physiological responses in the plants. Their ability to cause an increase in length of axis is used to increase the length of the grape stalks. Gibberellins causes fruits like apple to elongate and improve its shape. They also delay senescence. Thus, the fruits can be left on the tree longer as so as to extend the market period. GA3 is used to speed the malting process in brewing industry. Sugarcane stores carbohydrates as sugar in their stems. Spraying sugarcane crop with gibberellins increases the length of the stem, thus increases the yield by as much as 20 tons per acre. Spraying juvenile conifers with GA3 GS hastens the maturity period, thus leading to early seed production. Gibberellins also promotes bolting, internode elongation just prior to flowering in beet, cabbage, and many plants with rosette habit. 15.4.3.3 Cytokinins. Cytokinins have specific effects on cytokinesis and were discussed as kinetin, a modified form of adenine and a purine. From the autoclaved herring sperm DNA, kinetin does not occur naturally in plants. Search for natural substances with cytokinin like activities led to the isolation of zeatin from corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occurs. For example, root apex, developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient mobilization, which helps in delay of leaf senescence. 15.4.3.4 ethylene. Ethylene is a simple gaseous plant growth regulator PGR. It is synthesized in large amounts by tissues undergoing senescence and ripening fruits. Influence of ethylene on plants include horizontal growth of seedling, swelling of the axis, and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plant organ, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. This rise in rate of respiration is called the respiratory climactic. Ethylene breaks the seed and bud dormancy, initiates germination in peanut seeds. Sprouting of potato tubers, ethylene promoters rapid internode petiole elongation is deep water rice plants. It helps leaves, upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapple. It also includes flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethophone. Ethophone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Ethophone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnut, it promotes female flowers in cucumbers, thereby increasing the yields. 15.4.3.5 Abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy. But like other PGRs, it is also has other wide range effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. ABA inhibits seed germination. ABA stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore, it is also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA helps to seeds to withstand decision, desiccation, and other factors unfavorable for growth. In most situations, ABA acts as an antagonist to GA, gibberellic acids. We may summarize that for any and every phase of growth, differentiation and development of plants, one or the other PGRs has some role to play. Such roles could be complementary or antagonistic. They could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect the event. Example, dormancy in seeds, buds, abscission, senescence, apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control along with genomic control and extrinsic factors. They play an important role in plant growth and development. Many of the extrinsic factors, such as temperature and light, control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factor on initiation of flowering. 15.5 Photoperiodism. It has been observed that some plants require a periodic exposure to light to induce flowering. It is also seen that such plants are able to measure the duration of exposure to light. For example, some plants require the exposure to light for a period exceeding a well defined critical duration, while others mu- must be exposed to light for a period less than this critical duration before the flowering is initiated in them. The former group of plants are called long day plants, while the later ones are termed short day plants. The critical duration is different for different plants. There are many plants, however, where there is no such correlation between exposure to light duration and induction of flowering response. Such plants are called day neutral plants. Figure 15.12. It is now also known as that not only the duration of light period but that of the duration of dark period is also equal importance. Hence, it can be said that flowering in certain plants depends not only a combination of light and dark exposure but also their relative durations. This response of plants to periods of day night is termed as photoperiodism. It is also interesting to note that while shoot apex modify themselves into flowering apex prior to flowering, they, that is, shoot apex of plants by themselves cannot perceive photoperiods. The site of precipitation of light, dark duration, or the leaves. It has been hypothesized that there is a hormonal substances that is responsible for flowering. This hormonal substances migrates from leaves to the shoot apex for including flowering only when the plants are exposed to the necessary inductive photoperiod. 15.6 Vernalization. There are plants for which flowering is either quantitatively or qualitatively dependent on exposure to low temperature. This phenomenon is termed as vernalization. It prevents precocious reproductive development late in the growing session and enables the plant to have sufficient time to reach maturity. Vernalization refers specially to the promotion of flowering by period of low temperature. Some important food plants, wheat, barley, rye, have two kinds of varieties, winter and spring varieties. The spring variety are normally planted in the spring and come to flower and produce grain before the end of the growing season. Winter varieties, however, if planted in spring, would normally fail to flower to produce mature grain with a span of flowering season. Hence, they are planted in autumn. They germinate and over winter come out as small seedlings. Resume growth in the spring and are harvested usually around midsummer. Another example of vernalization is seen in biennial plants. Biennial are monocarpic plants that normally flower and die in second season. Sugar beet, cabbages, carrots are some of the common biennials. Subjecting the growing of a biennial plant to cold treatments stimulates a subsequent photoperiod flowering response. 15.7 Seed dormancy. There are certain seeds which fail to germinate even when external conditions are favorable. Such seeds are understood to be undergoing a period of dormancy which is controlled not by external environment but are under endogenous control or conditions with the seed itself. Impermeable and hard seed coat, presence of chemical inhibitors such as abscisic acid, phenolic acid, para-ascorbic acid, and immature embryos are some of the reasons which causes seed dormancy. This dormancy, however, can be overcome through natural means and various other man-made measures. For example, the seed coat barrier in some seeds can be broken by mechanical abrasions using knives, sandpaper, etc. Or vigorous shaking. In nature, these abrasions are caused by microbial action and passage through the digestive tract of animals. Effect of inhibitory substance can be removed by subjecting the seeds to chilling conditions or by application of certain chemicals like gibberellic acid and nitrates. Changing the environment conditions such as light and temperature are other methods to overcome seed dormancy. In summary, growth is one of the most conspicuous events in any living organism. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involved increased protoplasmic material in plants, meristems, or the sites of growth, Root and shoot apical meristems, sometimes along with intercalary meristems, contribute to the elongation growth of plant axis. Growth is indeterminate in higher plants following cell divisions in root and shoot apical meristems cells. The growth could be arithmetic or geometrical. Arithmetic or geometrical growth may not be and generally is not sustained at a high rate throughout the life of cell, tissues, organism. One can define three principal phases of growth: the lag, the log, and the senescent phase. When a cell loses the capacity to divide, it leads to differentiation. Differentiation results in development of structures that is commensurate with the with the function the cells finally has to perform. General principles for differentiation for cell, tissues, and organs are similar. A differentiated cell may de-differentiate and then redifferentiate. Since differentiation in plants is open, the development could also be flexible. That is, the development is the sum of total and differentiation. Plant exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factors. Intercellular intrinsic factor are the chemical substances called plant growth regulators (PGR). There are diverse group of PGRs in plants, principally belonging to five groups: auxins, gibberellins, cytokinins, abscisic acid, and ethylene. These PGRs are synthesized in various parts of the plant. They control different differentiation and developmental events. Any PGR has diverse physiological effects on plants. Diverse PGRs also manifest similar effects. PGRs may act synergistically or antagonistically. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such external factors. Flowering in some plants is induced only when exposed to certain duration of photoperiod. Depending on the nature of photoperiod requirements, the plants are called short day plants, long day plants, and day neutral plants. Certain plants also need to be exposed to low temperature so as to hasten flowering later in life. This treatment is known as vernalization. Thank you.